Hi everyone, my name is Chanelle Webster. Thank you for the introduction, Paul. I'm here to speak to you today about my honours project completed last year that looked at the effect of an extreme summer rainfall event on the resilience of seagrass species. Of an estuarine seagrass species rather. So before we get into the project, the main stressor we're concerned with here is salinity. So salinity regimes around the world are changing under different stresses such as climate change and extreme rainfall. Under climate change, increasing temperatures and declining precipitation are causing gradual increases in salinity, and I'm focusing on estuarine ecosystems which have somewhat restricted circulation. So on the one hand, you have gradual increases. Under extreme rainfall events, however, these cause rapid declines in salinity. So in terms of a global context, where could this be an issue? In Mediterranean-type ecosystems, which have hot, dry summers and cool, wet winters, this could be an issue in terms of how it affects seagrass resilience. I will specifically be focusing today on a system in southwestern Australia. So just a little bit more from the resilience perspective, resilience being the capacity of a system to resist and recover from disturbances. What we might be interested in understanding is the timing of the event, the magnitude and duration. Particularly in summer, when background salinities are high, when these extreme rainfall events come through, they cause the salinity to decline, which means that seagrasses have to tolerate a higher magnitude of change in salinity. Whereas in winter, that magnitude would be that much smaller. Of course, we might also be interested in the duration at which the seagrasses experience those lower salinities. So I'll provide some site background for you. The system that my research was focused in is the Swan River Estuary located in temperate southwestern Australia. It's a permanently open estuary, the dominant seagrass species being Halophila ovalis. So the seagrass distribution map here shows you where seagrass meadows occur throughout the system. The main growing period is between December to March, when salinities are high, temperatures are warm, and light conditions are fairly good. These meadows support important faunal species such as the black swan. So the extreme floodfall event came through in February. What I'm going to do now is show you an animation of how salinity changed over that period. So you'll see the date stamp down the bottom here, the 7th of February. The majority of the rainfall came through on the 10th. What I'd like you to do is focus on this top graphic, and the reason why is because seagrass meadows that are monitored in the system occur in shallow areas of the system. So if you could just focus on that one, what you'll see here also is this graphic is shown in summer, so when the system typically experiences high salinities. Yeah, it's working. So the majority of the rainfall occurred here in the catchment system. And you can see a decline in surface salinity throughout the whole system there. What you see here is that opening towards the ocean. So I'll just provide now a little bit more background on mean rainfall that's shown along the y-axis here. The months during summer, so December to March. The long-term average, so from 1993 to 2018, demonstrates that rainfall typically is fairly low during the summer months. You'll see two extreme rainfall events that have occurred in the system here. The first in 2000 and the second in 2017, where the year in which my study took place. So from a seagrass monitoring perspective, in 2000, when that first event occurred, there was no monitoring program established in this system. So the impacts to seagrass could only be inferred. In 2011, however, the Department of Water and Environmental Regulation began monitoring seagrasses in this system. What you'll see, the bottom right-hand side here, is the flood plume that followed the February rainfall event. The green stars indicate the 6 seagrass meadows that are monitored by the Department of Water between December to March. So in 2017, when this flood occurred, it provided an opportunity to understand how seagrass resilience may be affected. And this is where my project comes in. So metrics have been used to understand different aspects of marine heat waves, such as their intensity or magnitude, but not yet for extreme rainfall events. Halophila ovalis, the dominant species in the system, has been shown to be sensitive to prolonged hypersalinity conditions, seeing effects such as reduced growth, which could impact its resistance capacity. So my project had 2 aims, the first being to quantify different characteristics of the flood, such as its intensity and duration, and the second aim was to determine how the resilience of the dominant seagrass species Halophila ovalis was affected by this event. In terms of our aims, the metrics that I've mentioned, this schematic here shows these metrics being developed in a marine heat wave scenario. So I won't spend too much time on this, but one of the characteristics you might be interested in is what is the peak temperature that's reached in terms of difference from that background temperature during a heat wave. What I'll instead focus on is these metrics being applied in the extreme rainfall scenario. So on the y-axis, you've got salinity, and along the x-axis, the day of the year, so time. Background salinity shown in blue, and Imax refers to the difference between background salinity and the lowest salinity that was reached following the extreme rainfall event. In order to calculate these metrics, we needed to, or specifically the duration, we needed to establish a discrete start and end date. So the definition we used was that the flood period would be the period during which salinity differed from background conditions by 5%. From that, we were able to determine when the flood started and when it ended at each site, and the duration would be the difference between that start and end date. Finally, the rate of onset would be the rate of decline in salinity to the lowest salinity that was reached during that extreme rainfall event. From the seagrass response side of things, so as I've mentioned, 6 seagrass sites were included in the study, and monthly sampling of different measures such as cover, biomass, leaf density, for example, were measured. What you see here is 2014 and 15, the general changes in salinity over summer and winter. These are years during which no flooding occurred. The orange line shows you the salinity changes at upstream meadows, and the salinity changes in blue are for the downstream meadows. So you can see that the upstream meadows experience a greater range in salinity compared to the downstream meadows. However, in summer, we expect that salinity range to decline because salinity is normally fairly high during the summer period. In 2017, when that extreme summer rainfall event occurred, you can see that the range of salinities that both upstream and downstream meadows were exposed to is much greater than that annual range that they typically experience. So that led us to hypothesize that given that upstream meadows experience a greater range in salinity, that they may be more resilient to the effects of an extreme rainfall event. So what did we find in terms of the flood metrics? What you'll see on the your left is the upstream meadow. So this is the background salinity during 2016 when no flooding occurred. So that black line there, both upstream and downstream. The magnitude of change in salinity, the red line showing you the change in salinity during the flood period, was greater upstream. So they were exposed to a greater magnitude of change in salinity. However, the decline in salinity was that much more rapid downstream. So what does this all mean in words? The highest magnitude of change in salinity occurred upstream. They also experienced the longest duration during which salinity differed from background conditions by greater than 5%. However, the onset of the flood was more rapid downstream. In terms of seagrass response, just focusing on total biomass shown on the y-axis, the months during which sampling occurred. On the x-axis, the red line shows you the month during which the flood occurred. This black line here shows you the average total biomass in previous sampling periods when no flooding occurred. We observed significant declines in total biomass, both upstream and downstream, within 1 month of the flood's occurrence. But the thing that seemed to stand out the most to us is that in the most recent sampling period, there appears to be some legacy effect where the downstream meadows have not returned to that background average, whereas you can see here the upstream meadows seem to have recovered. So that led us to the following conclusions. Upstream, despite the duration of the flood being longer, these upstream meadows appeared to be able to recover from extreme rainfall events of this magnitude. However, it's unknown what intensity or frequency of flood they can tolerate. Downstream, although the decline of salinity was more rapid, and this could have impacted their ability to recover. So these meadows appear to be less resilient to extreme rainfall events of this magnitude, and it's unclear whether they will persist under more intense events. That leaves them in a vulnerable position to ongoing disturbances. The metrics that we used can be used to understand the impacts of extreme events on seagrass resilience, and now to facilitate comparisons with subsequent events. So how to answer the sorts of questions, how the intensity of salinity may be changing in between future events. So a little where to from here, I'm now currently doing my PhD with an ongoing resilience and estuarine focus. One of our plans is to simulate the flood and experimentally compare the upstream versus downstream response to see what role acclimation had with the results we observed. Other mechanisms that could influence seagrass resilience that I'm looking at is local adaptation and genetic diversity. So thank you all for your attention today, and to everyone who supported me during my research.
Thank you, Chanelle. Do we have time for one quick question before a tea break? Yes, please.
Great honors program.
Thank you.
Nice introduction to your PhD.
Thank you.
I'm just curious that when you showed us the changes in the aerial photographs of the flood, there was obviously looked like a lot of turbidity associated with it. So can I ask you why you've chosen salinity other than other stressors such as turbidity? Is it easy to measure, or do you think the turbidity wasn't enough? Or is it just covariance? Or do you think salinity is the stressor, or could it be turbidity?
Okay. I'm really glad that you did ask that question. Because I do believe that other stressors could have contributed to the decline. However, we looked at the correlation between salinity and other water quality parameters, such as Secchi depth, and the correlation was quite strong. What we feel is because there was such a magnitude of change in salinity, that at least in the short term, the salinity would have been the main stressor that produced the decline. But certainly, I mean, this is shown maybe 2 weeks after the flood. It could have had an impact. We did, however, see the leaves drop as well, which could indicate some stress response to those low salinities that were reached. Yeah.
It's a follow-up. Do you expect if this continues on, you may get a change in the actual species itself? To almost say like Rupia, which is known to actually be able to handle greater ranges of salinity. Was it the adaptation side of things?
So Rupia does actually occur in the system that I worked in. Whether there's going to be any shift in species composition certainly could occur. Yeah. Thank you.
Okay, thank you all. Let's put our hands together for all the speakers this afternoon.